discussing about the probable ancestors, and we had seen that right from the algae to gymnosperms, almost each and every group has been tried as a probable ancestor. And so when we are studying these different theories, we have to compare them. So what are the similarities? What were the stands? What were the basis? And obviously, after the study, what has outcome? And so whether the theory has been accepted or rejected. Even though there are large number of theories which have been forwarded, right from the algae to gymnosperms, and almost all the groups from gymnosperms, like cycadans to nettles, there are very few very close ancestry and similarities were found in some groups, and because of that, certain theories they became more popular. We had studied the two theories yesterday. One was the nettles theory, which is the existing group now, and which is the most advanced group among the gymnosperms. Also, we have studied the Benititles group or Cycadioidea group, and this Benititles or Cycadioidea, this is a totally extinct group which was formed at the beginning of the Mesozoic period, that is at the Triassic period somewhere. However, because of certain unknown reasons, this group could not survive further. It became extinct. And this Benititles group is considered as one of the most primitive group among the gymnosperms. So obviously, as this group was also showing, the bisexual strobili, which was then termed as a flower, obviously several scientists they tried to compare this Benititles group as a probable ancestor with the Angiosperms. However, we have seen that both nettles and Benititles, even though they belong to the gymnosperms, they are typical gymnospermous characters. Especially the wood character is totally of gymnospermous, which is having a large pith and obviously which is having a very thick cortex but very small vascular bundle or very small size of the vascular tissue. And this particular character is not presented anywhere. It is not present anywhere in the angiosperms. As far as the development of the xylem is concerned, in gymnosperms, the pitted thickening and the bordered pitted thickening both are most very common prevailing forms. Whereas in case of angiosperms are concerned, the scalariform thickening, reticulate thickening, and other forms, they are prevailing. So obviously from anatomical point of view, both are seem to be evolved independently. And obviously we do know that most of the gymnosperms, they are having the unisexual flowers or cones, and the cones may be, the plants may be either dioecious or monoecious. And because of that, there are several differences there in between the gymnosperm group and angiosperm group. Obviously, in the gymnosperm, the seeds are present, but the fruits are not there. And because of that, the formation of seed and the formation of ovule in angiosperms, it cannot be compared at the parallel level. And because of that, this theory, both nettles theory and Benititles theory, they ultimately were rejected, and it was assumed that both nettles and Benititles, on the other hand, they have shown a parallel course of evolution in the due course, and that's why they were showing the similar characters. And as we were discussing right from the day first, when we were trying to seek or find out the time of the origin of the plants, several references which were present in the Cretaceous period of the fossil angiosperms, they were belonging to the extinct genera. Extinct genera means the genera which were present at that time, but those belong to the advanced angiosperms. Even the records which were present of the dicots and monocots, they were showing the clear typical characters of dicots and monocots together, including the stomata, the venation pattern, and many other characters. And because of that, the paleobotanists, they were very serious about the fact that they are thinking that Cretaceous period cannot be a period of origin of angiosperms, but on the contrary, we have to go still below, still back into the time, and we have to seek somewhere where there has to be a very simplified form of the Strobilus. And obviously, as a large number of records were available, particularly with the microspores and megaspores, obviously the fronds and the wood characters, it was assumed that the firm identification of the fossils, which was made in even Permian period, Carboniferous period, there was a question mark. Some taxonomists they assumed, or paleobotanists assumed, that they were not the fossils of pteridophytes, but they were the fossils of the angiosperms. On the contrary, the microspores which were reported at that time during the Permian period, many of the paleobotanists they assumed that these were the microspores of the angiosperms because they were having the monocolpate or tricolpate structures. Unfortunately, both these forms they were present even in the pteridophytes. And today also in angiosperms we will find that almost half of the genera in angiosperms they show the monocolpate form, and remaining primitive forms they show the tricolpate form. Tricolpate means the microspore is having the three ridges on its surface, and it indicates that they have been developed because of the tetrasporic development of the microspore. And because of that, the dominant group which was existing in the Permian period is somewhere about 280 million years ago, and before that, it is in the Paleozoic period. So many of the paleobotanists, they were thinking that we have to find out the ancestry of the angiosperms with that of the pteridophytic group. Now, in the 18th century, the paleobotanists, they were much more happy and surprised because they found certain fossils where the fossil were showing the occurrence of intact seed on the frond. It was very interesting finding that a fern plant was present. Obviously, we do know the ferns, and what the ferns are having, they are the sporophytic plants, and the fern plants, they are having the independent separate gametophyte. So gametophytic prothallus is separate, but obviously in pteridophyte itself, we find that there is an extensive reduction in the gametophytic phase. And obviously in Selaginella and in other plants like lycopods and many others, we have seen that the gametophytic phase is totally reduced from exophytic form to endomytic form. Endophytic means the gametophytic phase is developing within the spore. So the female gametophyte was present in the megaspore, where the megaspore was still present in the megasporangium, and the megasporangium was still present on the megasporophyll. That is what we call as a sori. And the sori were present on the fern leaf. So this was called as endosporic development of gametophyte, which was a one step ahead, one leap ahead, which had been shown by the very rigorous development of evolution by the ferns. Obviously, when some records were identified, these records, they were having the seeds which were attached to the fronds. And that's why this particular group, which is not existing today, they gave a new name to this group, and this group was called as the Pteridosperms. That is the pteridophytes which are bearing the seeds on their body. And so this was again a sort of a distinct separate group from the pteridophytes, and it was a sort of a link between the pteridophytes and gymnosperms. And so As early as in 1947, Andrew, he proposed a hypothesis that the pteridosperms or the seed forms which existed in the Permian period, who existed in the Devonian period, who exist and who were dominant in the Carboniferous period, they have been considered as the ancestors of angiosperms. Yesterday, we were discussing this issue when Dr. Patil Madam has asked a question about why there was large number of fossil records are present of the pteridophytic groups or pteridophytic fronds in the coal mines. Obviously, Carboniferous period is the period which is almost 320 million years ago. So during the same period, this particular advanced pteridophytic group was also existing. And this group is now called as a Pterosperms group. Pterosperms, they are considered now as ancestors of angiosperms by a large number of paleontologists like Andrews 1947, Arnold 1949, Cronquist 67, and this was very thoroughly advocated even by the Takhtajan 1969. From the Paleozoic era, with the Upper Devonian period, Upper Devonian period is somewhere about 340 million years ago, to Permian period, that is 280 million ago. So this is a very large span of almost more than 60 million years. And this region, it was having or it was showing this particular group which was present. So this was at that time an advanced group. What it group had? So this group was showing the leaves which were similar to that of pteridophytes. So the leaves were very similar to that of pteridophyte. Pteridophyte means they were having the fronds. So you know the frond. Frond is nothing but it is having a petiole, it is having a leaf base, it is having the axis, and on the axis the leaf lamina, they are dissected. And each and every leaflet is having a prominent midrib. In case of angiosperms, what we are having a single leaf, but here there is a frond which is present. Now these leaves, they were associated with a cycad-like stem. Now remember, when we talk about the cycad-like stem, here it is expected the external morphology. Means what? You know the example of cycas. On the cycas, on the outer side, there are leaf scars are present. So you can see the leaf scars on outer surface. And that's why from external side, this stem looks like a very similar to cycas. Similar example is present with us also. We are also having the cycas with us, where the stem is having the leaf scars. Another best example of such a type of example is in the monocots, where in the monocots, particularly in the palms, we are having the leaf scars. If you see the palms, for example, if you see the coconut, if you see the bichel, if you see even the palms and other forms, सगळ्यात महत्त्वाचं म्हणजे आपल्याकडे शिंदी. शिंदी झाड बघितलं असेल, सगळ्यांनी बघितलं असेल आपल्या आसपास, अशी खेडेगावामध्ये सापडतं ते रस्त्याकडे असतं. तर त्याच्यावरती तुम्हाला दिसतं की त्याचा जो प्रत्येक लीफ आहे, तो कंपाऊंड लीफ आहे, त्याचा स्टॉक जो आहे, लीफ बेस जो आहे, तो पानापासून किंवा त्या खोडापासून बाजूला होत निघत नाही, तो त्याला चिटकून राहतो. So that particular format, that is called as a cycad-like stem. So on the basis of anatomical structure, they were considered as a distinct group from the ferns. And obviously this group was treated as an advanced group of pteridophytes. Later, when this group was discovered, that is the fronds, when they were discovered with the seeds together, they had been named variously at the time, like cycadophylicals. Cycadophylicals means they are having the stem like cycas, and they are having the leaves which are similar to filicales, means ferns. So the leaf-like ferns which are present on the cycas-like stem. Or also they were called as Pteridosperms. Pterido means obviously ferns, and sperm means seeds. So the seed-bearing ferns, that was another name which became more popular there. And even in sometimes in simple words, they are called as the seed ferns also. Now this is a reconstruction of a fossil records. Now remember, when we find the fossil records, we have never found a single plant as such. On the contrary, we will getting the records of each and every part independently. So what we are seeing here is nothing but It is a reconstruction. So we are bringing all the compounds together and then we are creating this plant. So this is a totally artificial image. Remember, when the fossils are identified or when the fossils are formed, the fossils are given the names based on their characters. So obviously, if the dendron is present, it means it is a stem character. If filicales is present, it is meant by it is a leaf character, and so on and so forth. So because of that, in the left side figure also, you will find that different names of the genera have been given. Some genera are the names of the fronds, some genera are the names of the stem, and even there are names even for the roots also, the names for the seeds also, the names for the reproductive bodies like the male cone and female cones also. Means, let us come to the right side image. Now in the right side image, you can see at the bottom a frond-like structure. Actually, it is a photograph of a fossil. It is a frond-like structure and it is showing very similar to the leaf lamina, small leaflets, what we call the leaflets in the ferns. And they are very similar to what we today know as the Laginopteris. So we are knowing the Lagonium today. आपण लायकोडियम बघतो किंवा आपल्याकडे चिलांथस पाहतो, याची पानं त्याच्यासारखीच आहेत. So this was given another name, the stem was given another name, and now you can see at the right side top there is one image which has been created, and that image is nothing but it is a megasporophyll which is containing, that megasporophyll is called the cupule. It is containing a single megaspore. That megaspore is containing the ovule, and this megaspore is covered by the sterile megasporophylls. That's why this is called the cupule structure. So this is nothing but a sort of a seed character which has been reported, which is associated with the Pteridosperms. Now this theory of the Pteridosperms, and they are the probable ancestors of the Angiosperms, is based on the following characters, and it is sufficient to bring the Pteridosperms and the seed ferns or the seed ferns closest to Angiosperms. On the basis of the stem, leaf, and seed characters, these have been regarded as the probable ancestors of even cycadales and benititales also. Because obviously when the cycadales and benititales evolved, they were evolved even from the Pteridosperm, because Pteridosperms were present much more earlier. Now the exhibited characters, these Pteridosperms, they showed the characters like reticulate venation, which is present in the dicots today. They showed the monopodial branching. For example, in pteridophytes, you know that there is a dichotomous branching. That is, each time a main axis splits into two, and the two cells, they start growing together. Like the example of a Rixia, the several examples even of the pteridophytes, which are showing a dichotomous branching. Here we are getting a monopodial branching, means only one axis continues to grow further. That is called as a branch of unlimited growth, whereas the other branch, it always remains short. तर तुम्ही आपल्याकडे बघितलं असेल, आपल्या अँजिओस्पर्म्स मध्ये, आपल्या डायकॉट्स मध्ये, इव्हन आपल्याकडे फर्स मध्ये एकच फांदी सरळ वाढत राहते, दोन फांद्या वाढत नाही. तो असा व्ही तयार होत नाही, एकच फांदी वाढते आणि दुसरी फांदी त्याचं मॉडिफिकेशन होतं. It gets modified either into the stipule, it may get modified into the frond, or it may get modified into the obviously reproductive body, that is a female cone or a male cone. And that's why the monopodial branching is a very interesting character, which is very common in both pteridophytes as well as in angiosperms. So remember, such a type of character is not present in the gymnosperms. In gymnosperms, we are having a polypodial branching. दोन branches वेगळ्या तयार होतात ना. Also, there is one more advantage, and that advantage is that there is a presence of cambium. Now, this cambium, when it is present in the stem, the cambium is producing the secondary growth. Remember, the pteridophytes, pteridosperms, and angiosperms, they are homologous by their structure, internal structure. So what is this internal structure? In pteridophytes, in pteridosperms, and in angiosperms, we are having the large vascular bundle, very small cortex, and very small pith. Means the wood is made up of secondary growth. The wood is made up of secondary wood. And because of that, this is the character which is even seen in pteridophytes, it is seen in pteridosperms, and the same character is existing today also in all the dicots. What we see that the secondary growth is present, that the vascular tissue is more, the pith is very small. Even the epiblema, or what we call the cork, is also very small outside. म्हणजे झाडाची तुलना किंवा बुंद्याची तुलना केली तर त्याच्यावरचा कॉर्क जो आहे, ज्याला आपण बाहेरची बाजू म्हणतोय, साल म्हणतोय, ती अत्यंत पातळ असते आणि त्याच्यामध्ये किती बाबतीत असतच नाही मध्ये. आणि हा सगळ्याचा सगळा खोडाचा भाग जो आहे, हा सगळ्याचा सगळा सेकंडरी ग्रोथ आहे. इट इज सेकंडरी व्हॅस्क्युलर टिश्यू. And the same structure is found even in the pteridosperms and even in the pteridophytes. However, the cycadales group, Benititales group, and even today the Coryphates group, they show a different type of wood character. In that case, the pith is large. What is the pith ultimately? Pith is nothing but it is a parenchymatous tissue which is depositing the secondary material. For example, it is depositing the tannins, it is depositing the gums, it is depositing the resins, and that's why it is black in color. So in gymnosperms, the vascular tissue is less, vascular cylinder is less, but it is having a very thick core. And this thick core is because of, again, the permanent persistent leaf bases. When the leaf bases they get attached, they cannot be detached from this place, because such a type of detachment character, what is known as an abscission, such a type of abscission is seen only in case of angiosperms. That's why in angiosperms, the leaves after certain time they get detached. म्हणजे पानं गळून पडतात आणि पानं गळून पडण्याचं कॅरेक्टर जे आहे अँजिओस्पर्म मध्ये आहे. परंतु पानं गळून पडायचं कॅरेक्टर हे जिम्नोस्पर्म मध्ये कुठेही नाही, हे पाम्स मध्ये कुठेही नाही. तुम्ही असेल तर पामची ती पानं कायमची चिटकून असतात. झाडं मारायला आले की मग ती खालची पानं गळून पडायला लागतात छोटी छोटी, तोपर्यंत ती झाडं ती पानं येतच असतात मेन स्टेम वरती. सो दॅट इज वाय फ्रॉम स्ट्रक्चरल पॉइंट ऑफ व्ह्यू, anatomical point of view, the angiosperms are much more closer to the pteridophytes and pteridosperms. The structure of the ovule of the pteridosperms, cycadales, benititales, and angiosperms, because all these are seeds ultimately, they consist of two basic parts. Obviously, one is a megasporangium, or this is also simply in Latin it is called the nucellus, because this megasporangium is containing ultimately the megaspore, and this megaspore will be ultimately giving rise to, after the meiotic divisions, it will be giving rise to the female gametophyte. You remember, when we talk about the megasporangium, we are talking about a reproductive body. It is still a part, a sporangium, it is a part of the sporophyte. That's why it is called the nucellus. And then this megasporangium, which is further going to bear the megaspores, that is gametophyte, they are generally surrounded by the protective coverings, and these protective coverings are called as integuments, that is walls. So remember, in angiosperms also the ovule is having two walls, outer wall and inner wall. Similarly, in other pteridophytes also there are two walls are present, outer and inner wall. However, in Nitales, the ovule is containing three walls, so it is tritegmic. Even in conifers, the ovule is tritegmic. In angiosperms, it is bitegmic. So the morphological nature of the nucellus, as established by the Hofmeister, is nothing but it is having a same analogy, homogeneity in all these groups. That is, the megasporangium is nothing but it is a nucellus which is going to give rise to the archegonium, which is going to contain the egg at the end. The structure of integuments has been variously interpreted, but the most acceptable theory has been given by the Benson, and obviously it has been modified to some extent by other workers also in 20th century. According to this theory, the ovule is nothing but it is a synangium in which all the sporangia except the more or less central one, only one sporangium, that becomes functional, and all other remaining surrounding ones, they become sterile. And that's why they have taken up the function of a protection. Means in the megasporangium, when all are functional, what is going to happen? All are going to produce the megaspores. But here in synangium, the best example of the synangium obviously is the synangium, is the xyloton. In xyloton, the three synangia are present. And what is happening here? Only the central megasporangium that is remaining functional, it is going to produce the megaspores, whereas the remaining other which are surrounding to this one, they are becoming sterile. They are not producing the megaspores. That's why they are ultimately, they are taking up the function of the protection. So ultimately these are nothing but they are the integuments. So according to Benson, the ovule is a synangium in which all the sporangia, obviously it is a megasporangia, except more or less the central one, it has become sterile. The outer have become sterile, and they have taken up the function of a protection, that is integuments, for the fertile sporangium. And this can be traced for their synangial origin. And this particular synangium, which is containing one single central fertile megasporangium surrounded by sterile megasporangia, this was called as a cupule. A new name was given, that is a single ovule or seed which is containing the sterile structures. It was called as a cupule. I suppose I have shown you the image of the cupule. You can see this image which is at the right side, at the top side. You can see a central ovule which is surrounded by the sterile megasporangia. The mega and microsporophylls, they are often born on the same plant. So obviously the plants were dioecious. Male and female cones were separate, but they were born on the same plant. But they were not arranged in a same definite strobili. Remember, in pteridophytes and in pteridosperms, the microsporangia, they were usually born on the axial side, axillary side of the fronds, whereas the female megasporophylls, they were present and they were born at the center of the cone or the central of the stalk or central of the stem. So that was the major difference. I will give you a very simple example so that you can understand. In the nephrolepis, the microsporangia or sori, they are present on the fronts and they are present towards the lower side. Whereas in case of cycas, the female cone is present at the center and it is coming from the central location. So that's why there are two locations which are totally separate. Generally, the microsporangia, they are present in the axillary position, and the megasporophylls, they are generally present in the terminal position or the central position. Now you can see the single image which has been reconstructed, which is indicating the seed habit. Now the name was given Legionopteris. Legiono means seed, pteris means the fern. So fern which is bearing the seed. Now you can see the right side image, and this right side image shows the presence of a single megasporangium, that is a nucleus, that is a nucellus. This nucellus is having its own walls, that is integuments, and these integuments at the center, you can see they are having the female gametophyte. And this female gametophyte, it is containing the archegonia. You can see the label at the bottom, the mega gametophyte with the archegonia. So the archegonia are present, and these archegonia, they are having a column, a cavity, and this cavity was nothing but it was a micropylar cavity or archegonial cavity. This cavity was opening outside so as to catch the microspores. And you can see the central megasporangium is surrounded.
Excuse me, sir.
Yes.
Sir, your presentation slide cannot be visible.
This is नहीं है.
नाही सर, तुम्ही बोलता तेच फक्त दिसतंय, कम्प्युटरची स्क्रीन नाही दिसत आहे.
एक काम करा, मी पुन्हा प्रेझेंटेशन करतो. बाकी सगळ्यांना दिसतंय का?
येस सर.
बाकी सगळ्यांना दिसतंय?
नाही सर.
येस सर.
कुणाला किती लोकांना दिसतंय? एक काम करा, मी पुन्हा लॉग इन होतोय. मी प्रेझेंटेशन पुन्हा करतो, हे स्टॉप करतो आणि परत चालू करतो. ठीक आहे? मी परत स्क्रीन शेअर केलेला आहे. आता दिसतोय का?
येस सर.
हॅलो, कॅन यू सी?
येस सर. येस सर.
ओके सर. बरं, ठीक आहे. मागच्या जे एक दोन स्लाइड होत्या पुन्हा रिपीट करेन नंतर. आता आपण पुढे जाऊया. कॅन यू सी द इमेज नाउ? Now this is the image of the seed where the megasporangium is present at the center. This megasporangium is containing a single megaspore, and these megaspores, they are giving rise to female gametophyte, and this female gametophyte is containing the archegonia. And that's why this is a endosporic female gametophyte, and this particular megasporangium is surrounded, that is nucellus, is surrounded by the integuments, walls. And outside again, you can see the other additional walls, which is known as a cupule. Now, this cupule is showing some cupule glands outside, so that the insects at that time, they were attracting the insect forms, and because of that, the pollination was visible and possible. The best evidence for this hypothesis is afforded by the ovules of the Pteridosperms, which is seen in many well-deserved forms in all the groups. A similar explanation also applies to the ovules of other groups of the plants, that is in Gymnosperms as well as in Angiosperms. That is, in all these cases, except in Gymnosperms, the ovules are directly born. Whereas in case of angiosperms, the ovules are born again in another putative layer, which is known as the ovary. And that's why as the ovules are enclosed in the angiosperms, they are called the angiosperms, where the ovaries are enclosed. Ovules are enclosed. In other words, the ovules of all other groups of the seed plants, they have probably a common structural basis and ancestry. That is, they are following a typical common pattern which is seen in all other groups, which has begun from the pteridophytes, and where it was called as the seed-bearing ferns. According to the Long, in 1966, the first integument of the angiosperm seed is formed by the fusion of telomic units. Now, telomic unit is nothing but, remember, this is the nucellar part. That is the part of the female gametophyte which is providing the nutrition to the megaspores. But the second integument is an outgrowth either from the chalaza or from the first integument. What is the chalaza ultimately? Chalaza is nothing but it is a part of the megasporangium. So on one hand, we are talking about the megasporangium. In the megasporangium, there is a female gametophyte, and in the female gametophyte, it is containing the archegonia, and the archegonium is containing the egg. Remember, so ultimately, when we talk about the egg and when we talk about the ovule, Ovule is nothing but it is a female gametophyte which is present inside the female that is a megasporangium, that is nucellus. The similarities of the seed structure and the existence of one or several ovules subtended by a specific structure which is known as a cupule. Such a type of cupule, in case of angiosperms, the angiosperm carpel is considered as it is derived from the dorsiventral biwalled Pteridosperm cupule by this Benson, and accordingly he has put forwarded his theory. So what he has suggested is that in angiosperms also the ovule is nothing but there is a single fertile megaspore which is producing a female gametophyte, and then it is protected by the female megasporangia. Dear friends, let me tell you one thing, one major difference. And the difference is that in pteridosperms, in pteridophytes, and even in gymnosperms, the development of female gametophyte is a tetrasporic. Means after the division of the megaspore mother cell, 4 cells are formed, that is the 4 gametes are formed, which is called the megaspores. And all these 4 megaspores, they are functional in pteridophytes, pteridosperms, and gymnosperms. And that's why the female gametophytic development in these 3 groups, it is always called as a tetrasporic development. However, in case of angiosperms, within the 4 megaspores which are formed, only one becomes functional and all other 3 they are removed. They are disintegrating. And that's why in angiosperms, the female gametophyte or the ovule development is monosporic, not the tetrasporic. So this is the probable steps of seed evolution. You can see we have started right from the homogeneous ancestor, which is homosporous. That is, all the spores are equal. From there, the central image number 2, where it is written heterospory, where you can see again there are 2 cones, but these 2 cones, they are containing 2 different sizes of the megaspores and microspores. The microspores are small in size and they are many in number. Megaspores, they are large in size and they are less in numbers. The megaspores, they are further undergo next evolution where the megaspore is showing the reduction of the many megaspores into one megaspore only, and this one megaspore is germinating in the spore and the female gametophyte is present within the spore. Such a type of female gametophyte is known to us in Selaginella. This is present in even Lycopodium. So this is the next step of advancement where from many megaspores in a cone, only one spore is present in the cone, and the cone itself is containing the female gametophyte. Obviously, it is also containing the archegonium. We are going one step ahead, and here the megaspore which is present, it is present at the center, but that is surrounded by the integuments. Remember, these integuments are showing the female gametophyte at the center, which is containing the egg, and that is a sporangium developed by the integuments. So here, not only this cone is formed, but the cone is then protected by the advanced sterile involucral sterile megasporangia. And that's why this is a typical structure of a seed or cupule in case of pteridophytes. Cycadales or pentatales or any other known group of living gymnosperms cannot be considered as the ancestor to angiosperms. And thus, because of the elimination process, as these are not relative, by elimination process, seed pearls are now considered as the ancestors for the angiosperms by many authors. And this particular idea, it has become much more popular in 21st century. Even conquest he showed that the double fertilization and the development of triploid endosperm, this is again a further step in evolution which has been attained in the angiosperms, which is not even present in the pteridosperms. So Cronquist says that pteridophytes, they give rise to the pteridosperms which were containing a naked ovule, and this naked ovule was protected by the sterile megasporangia, and from there, from this ovule, the structure of ovary has generated in the angiosperms, and this structure of ovary, ultimately it is much more again advanced in evolution, where it is showing the double fertilization and obviously formation of post-zygotic formation of endosperm. Post-zygotic means the endosperm is formed in angiosperms after fertilization, whereas in case of pteridophytes, seed ferns, and even gymnosperms, the endosperm is formed before fertilization. And that's why in earlier members or earlier groups, the endosperm is haploid in nature because it's a part and parcel of the female gametophyte, which is haploid, whereas in case of angiosperms, it is triploid in case, where the diploid endosperm mother cell is fusing with one more set of genes which is coming from the microspore, and you know the embryology of angiosperms, which is giving rise to the triploid endosperm. Further, the origin of sepals from the leaves and petals from both leaves and stamens, these are the further course of evolution for the formation of a typical flower in angiosperms. So the angiosperm flower is nothing but it's a modified stem where leaves are modified into the bracts, and the bracts are further modified into the sepals, and even the sepals are further modified into petals, or even sometimes the microsporangia, that is obviously the stamens became sterile and they became petaloid, and they gave rise to the perianth, or they gave rise to the petals. Such a type of petaloid stamens, such a type of sterile stamens, they are present and they are represented in many families of angiosperms, and they are present in both dicots as well as in monocots. The most interesting evidence of the evolution of angiosperms comes from the fact that some primitive angiosperms, they lack vessels in the secondary wood, and this feature is also present in the seed ferns, that is the pteridosperms. So thus in pteridosperms the vessels are absent, in primitive angiosperms also the vessels are absent, and only the vessels are present in advanced angiosperms. Thus all the characters in which the angiosperms differ from the seed ferns could have evolved from the pteridospermous ancestry. According to Takhtajan, the angiosperms arose from a very ancient group of gymnosperms which must have the primary secondary xylem, a primitive secondary xylem of scalariform thickenings or brackets, at least in early wood, and which was having a primitive bisexual strobili. That is, the Takhtajan was claiming that the Benitites and primitive angiosperms, they must have got a common ancestry. From a common ancestor, these two groups must have evolved. However, this particular approach of the Takhtajan has not been accepted, and it is rightly been said that the wood of the angiosperms, leaves of the angiosperms, and the floral morphology of the angiosperms, it is more relevant and it is more close to pteridophytic group and not to the gymnospermous group. According to Melville, in 1960, he proposed a Golovkin theory, and he proposed that a reproductive branch of Glossopteridans, again a primitive fossil fern genera, a pteridosperm was somewhat very much comparable to the present day angiosperm Dicapetalum. Now remember, Dicapetalum is an existing genus of angiosperm belonging to the family Degeneraceae, which is very similar and close to Ranunculaceae and Magnoliaceae. And because of that, Melville, he has forwarded a hypothesis that Glossopterides is a probable ancestor of angiosperms, which gave rise to the Dicapetalum on one side, and from there the angiosperms might have evolved. However, there are two objections also which have been taken for the pterospermic ancestry. The first objection is that the absence of scalariform xylem vessels in the pterosperms, which is a general character, which is a common character in angiosperms. However, remember, scalariform thickening is present even in Xylotales group, that is in primitive pteridophytic group. Primitive means existing today, lower group, we can say lower, not primitive, lower pteridophytic group. On the contrary, in Pterosperms, the pitted thickening were present. So, unfortunately, no satisfactory explanation for the cupule as an equivalent of the capillary wall is recorded in case of multi-ovulate cupules. When we talk about the single ovule containing or single ovary containing a single ovule, this can be justified. But when we are talking about an ovary which is having the axial placentation, a family which is having the parietal placentation, at that time the number of integuments around each ovule, then that becomes a question mark for explanation. And that's why these are the two strong objections which have been raised by the people. And so today it has been accepted that all advanced groups of gymnosperms and angiosperms together, they all have evolved from a common primitive pteridospermous stock. That is, they have evolved somewhere much more earlier, even earlier at Ordovian period, somewhere 380 million years back. And from this seed habit form, all other modern groups, the existing groups, they have evolved, which are including, you can see the top, the most primitive, most lower group of gymnosperms, cycadales, and then coniferales, the jingo, and the Nitales group. Ultimately, it is leading further to the angiosperms. In spite of all these objections, the similarities which are found between the pteridosperms and angiosperms, they suggest that angiosperms have very close connections, relationship in embryological form, in morphological form, in developmental form, in ontogenical form with the pteridosperms. But however, because of the absence of any direct evidence, there is a growing tendency among the phylogenesis to accept the pteridospermic ancestry of angiosperms even at the present by rejecting all other claims. And that's why even though more than 16, 17 different hypotheses they have been forwarded, today the most valid one, the most strong one, and most compatible one is the ancestry of the angiosperms has to be found somewhere in the most primitive seed-bearing group which was present in the Carboniferous period and in Permian period, and that group was called as a seed points or pteridophytic ancestry. Ultimately, in conclusion, as it has been summed up by Pant and Kirwai in his book, it may be said that in spite of all the heroic attempts to solve the problem from various angles, there is a convincing evidence in support of either of the accepted time, nature of ancestral stocks, or the place of origin of angiosperms, and obviously a question mark problem which was raised by the Darwin itself, the origin of angiosperms still remains a question mark for the present generation of plant researchers. So this topic is not giving us any clear-cut claims, clues, and evidences, and because of that, all today's modern world taxonomists, paleobotanists, and even researchers, they are still working on finding out the probable place of the origin of angiosperms, probable time of the angiosperms, and probable ancestry of the angiosperms. So when we are talking about this and when we are coming to the conclusion, what the recent trend is saying, let me tell you one thing. This trend came up from the Bessay, who provided a Bessay's dicta, that flower. And then it was followed by the Smith, it was followed by the Cronquist, it was followed by the Crozite. And again, the same trend was accepted even in the advanced phylogenetic group. It has been now suggested that angiosperms should not be considered as a single group which has originated from a common stock. It has been now suggested that angiosperms, even though they are showing the similarity today, this similarity is because of the parallel evolution. Among the angiosperms, the different groups, they have evolved differently, separately, and there is no any correlation among oneself. So we are getting the trimerous dicots, that is the dicots which are having the trimerous flowers. We are having the tetramerous dicots, where the flowers are tetramerous. We are having the pentamerous dicots, where the pentamerous arrangement is present. Even we are having the hexamerous to octamerous arrangement. Even there is a superior ovary and inferior ovary, both are separate trains. Even we are having the monosulcate and polysulcate or trisulcate pollen grains, means some are having a single suture and others are having the three sutures or three radiate marks on the microspores. Even as per the embryo sac is concerned, many of the genera, 80% of the genera, they are containing the 8 nucleate embryo sac, but there are primitive angiosperms which are containing 4 nucleate embryo sac. There are some angiosperms which are containing only the stamens. There are some angiosperms which are containing the lamellar stamens. And there is one thing that now the scientists have accepted, that monocots are not evolved from the dicots. On the contrary, monocots and dicots, they are having a very long separate ancestry in the past, and they are showing a parallel evolution. So the gymnospermous group is totally separate, and even within the angiosperms also, the only character that the seeds are enclosed in the fruit, except this character, monocots and dicots, they are showing several characters which are dissimilar to each other. And that's why now the scientists have accepted that even the angiosperm group itself can be split. It has to be split into 2 separate domains. In one domain, there has to be a dicot group, dicot ancestry, and the dicot ancestry shows that within dicot, they are polyphyletic. The dicots also not evolved from a common ancestor, but they evolved to at least 4 to 6 different separate ancestors. So they are having the polyphyletic ancestry. However, in case of monocots, now the scientists have accepted today that they are having a monophyletic origin. That is, the angiosperms evolved, the monocots evolved from a single stock, single ancestor, whereas the dicots evolved from many ancestors. And then because of that, and because of the evolutionary trends, this particular question, this particular issue, to find out the origin of angiosperms is a very, really big question mark. And also this particular issue has not been resolved by the researchers. In the same way, why and how the human being only evolved on the earth, whereas all other animals are still grazing around us. They are not evolved, they are not changed. They are as it is, they were present almost more than 15 million years ago. So they have not gone any modification at all. But what has happened, why it has taken place, that within last just 10 lakh years, the human being, right from the apes, it is assumed that it has showed so much giant modification, evolution, that right from the monkeys, on one side the monkeys are also present and the human being is also present. So occurrence of human being on the earth is a big question mark. And in a similar way, the presence of angiosperms on a very largest scale with greatest diversity and maximum spread, it is also a big question mark. And that's why it is today, there is no any concrete answer for the time of origin, for the probable place of the origin, and probable ancestry of the angiosperms. So with this concluding remark, let us stop for the present. Today we have completed the origin of angiosperms. If at all you are having any questions, you are supposed to write those questions to me on the WhatsApp group, and tomorrow at 2 o'clock, if there are some questions, we'll be going ahead for the interactive session of discussion.